Okay, so hi, my name is Madeline Liddy, and I'm part of the group that designed a quantum aptasensor. So how our quantum aptasensor works is that we have two main stars of our sensor. The first star is a diamond that we grew on a silicon wafer. So diamond has this really cool defect in it called the nitrogen vacancy center. And this vacancy center has the ability that when excited with green light will emit red light. So that's really cool, but how does this actually work? How does it give us anything? So essentially what happens is we attached a DNA strand to the surface of our diamond, which has a complementary strand attached to it with a magnetic particle. So initially our setup starts with the magnetic particle very close to the diamond surface. And this results in a lower intensity of red light coming from the surface. Then we introduce our target protein. And what happens is that magnetic particle goes away because the DNA wraps around that protein. And that causes an increase in red light that we're able to see. So essentially what we're detecting is an increase or decrease in red light coming from our diamond. So because we have this ability, all you have to do is change the DNA that's on the diamond surface, and then you can detect a different type of protein. So this makes our sensor very unique because immediately you can see that we can detect any different type of biological protein, or you can detect a certain gene sequence, or you can detect certain other biological molecules. But the real cool thing is that you can actually detect and expand this to detecting any other small molecule. So all you have to do is basically change what's going on the diamond surface. So the next advantages of our system is that current sensors available can detect about micromolar ranges of target material. We're able to detect about 100,000 times more sensitive materials than that. So we're able to detect about 1 in every trillion particles. So that's one advantage, which is immediately able to understand. So another advantage is that typical setups that use the same kind of quantum mechanics associated with this project take up entire rooms, take years to build, and are incredibly expensive machines. So we were able to take something that's about the size of a room and shrink it down to the exact materials that you see on the table before you. So we shrunk it down in size. So immediately, higher sensitivity and decreased in size, decreased in costs, and increase in selectivity because we are able to detect any target molecule. So the applications for this, as I mentioned before, can be used for many different biological applications, including early signs of cancer detection, or early signs of heart disease, or early signs of pregnancy. Or in our case, we detected a certain protein that's associated with blood clotting. So, but you can expand this to water contamination detecting, or other trace materials that are found in the environment. So another thing we've proven is that we've able to detect in gas as well. So that opens up the realm of detection to things like particles in space, or you can detect toxic gases in mines, or toxic gases used in chemical warfare, things like that, making workplaces a lot safer. So essentially, this project is still proven that it works, and it's very, very simple in early stages, and upon future work can be taken to limitless applications and detection. So, thank you very much.